Okay, here we are. Thanks very much. Thanks everybody for joining this webinar and welcome. And so what I hope to be able to do over the coming 30 minutes is to present an overview of a technology that we believe is highly interesting and offers interesting prospects for unbiased genetic screening. But let me introduce you to the problem that we're facing. And the problem I want to address today is the fact that a lot of the drugs that we give to patients fail. And one of the reasons for failure is that the patients differ with regard to their genetic background, and it has an impact on how drugs perform in clinical trials. And ideally, you would like to be able to stratify patient populations into a group that is a non-responder group, and that group really doesn't benefit from the treatment, and a group that where the response rate is ideally 100%. So historically, people have tried to address the impact of the genetic background on treatment responses, and one way to address this is to work with cell lines. And so what people historically do to answer this question is they look at a large number of cell lines, drug sensitivity profiles, group the various cell lines into a sensitive group and a resistant group, and they try to find the mutations that confer resistance or sensitivity. And in an ideal world, that separation looks as depicted on that slide, where there is one group that has a mutation in gene A that, in that case, makes cells drug sensitive. However, in a real world, that mutation is occluded, if you like, by the presence of additional mutations that are confounding that analysis, and that make it very difficult to separate these groups and determine the underlying genetic changes that determine sensitivity or resistance towards drugs. And that is a particular problem in the area of oncology, where resistance to treatments, both primary and then later on acquired resistance, is a major limiting factor. And so what you would like to be able to do is, or what you would like to actually have at hand, is a system where you can study the action, the mechanism of action of a drug in a panel of isogenic cell lines. So these would be cell lines that are identical to one another with regard to their genetic background, but that differ in one mutation in one specific gene each, and have no additional genetic changes. However, that is something that's not entirely easily accessible. And so what's kind of missing in terms of establishing such a cell line, such an isogenic cell line panel, and to be able to do genetics in human cells, is illustrated on this slide, and that is that most of the cells that you're dealing with are, in fact, all human cells are, or most human cells are diploid. That means they have two genome copies, and as a consequence, if we introduce a mutation on one chromosome copy, the second chromosome copy will remain intact, and that is usually sufficient to mask the effect of the mutation. And so it turns out that there is one particular cell line in which this is not the case, and that is a cell line that Tijn Brummelkamp came across. So Tijn, back in the days, was working at the Whitehead Institute for Biomedical Research as a Whitehead Fellow, and he came across this very peculiar cell line that, whose karyotype is depicted on that slide. And as you can see, that cell line is haploid for most human chromosomes, with the exception of chromosome 8. Chromosome 8 is present in 2 copies, and a piece of chromosome 15 that is fused to chromosome 19. And so when Tyne saw these cells, he immediately realized that they offer a huge potential in terms of genetic screening, because every time you introduce a mutation in that particular cell line, the phenotype of that mutation would become immediately apparent. So if you like, this is really a paradise for a geneticist, because this is a human background. And so in the meantime, it turns out, and so obviously, you know, why is this paradise, or why is this such an ideal scenario? Again, illustrated on that slide. So in the diploid cells, again, we introduced the mutation, and the phenotype of that mutation is masked by the second copy that's intact. In the haploid cells, however, that mutation that we introduce will immediately, the phenotype of that mutation will immediately become apparent. And so it turns out that we do not only have access to KBM7 cells, but to two additional cell lines that were derived from them. So let's spend a few minutes on that panel of cell lines. The first one, again, is called KBM7. It was isolated from a chronic myeloid leukemia patient. It's near-haploid. So I mentioned the fact that it has 2 copies of chromosome 8, an additional piece of chromosome 15 appended to chromosome 19. It's a myeloid cell line, as you can see on this microscopic image. So these are suspension cells, and they have all the cell surface markers that you would expect from a myeloid cell. I can also say that these cells contain the Philadelphia chromosome. This is depicted down here, and they're actually imatinib sensitive. Then there is a second cell line referred to as HAP1, and these cells are very different. Although they derive from KBM7 cells, they're no longer suspension cells. Instead, they're adherent, and you can see on that microscopic image that they look a little bit like HEK293 cells. They're more haploid than the KBM7 counterparts. They've lost the second copy of chromosome 8, and we find them very suitable for a number of biological processes, and they clearly have display no myeloid markers anymore. And then the most recent addition to the haploid club, if you like, is a cell line called EHAP that we engineered back in the days at Haplogen, our Horizon Genomics. And that is a first somatic cell line that is fully haploid. It's derived from HAP1 cells. It's very similar in morphology. And this cell line is, if you like, the ideal model system to do genetics. And so what are the kind of screens that we can do in these cells? Conceptually, the genetic screens that we can do in these cells are very comparable to the experiments that people did in yeast 15 years ago. As you remember, yeast go through that haploid stage of the life cycle, and that allowed biologists to introduce mutations that would then later on persist in the diploid stage, and thereby essentially establish a large screening repertoire that you use to screen essentially any phenotype that you can establish in these cells. Now, how do we introduce the mutations? We use a technology that's called gene trapping. The gene trap in our case is delivered using a retrovirus, but other people have used transposons or lentiviruses. And the gene trap itself consists of a strong splice acceptor fused to GFP. And when that element is integrated, let's say in intron 1 of a gene, it abolishes splicing of exon 1 to exon 2 because of that very strong splice acceptor, and because it then also contains a polyadenylation signal, the mRNA will be truncated and gene expression will be disrupted. And so the beauty with this element is that we can deliver it with a retrovirus, and because we can generate very high titer retroviruses, we can generate populations of cells that essentially cover almost any gene that is expressed in these cells. And that is illustrated on the bottom part of this slide, where we start off with the haploid cells. We do the global retroviral gene trapping, end up with a population of cells. This is, several tens of millions of mutants. And then what we can do is we can apply selection. And the selection that we're talking about today is a compound treatment. So here we're treating this population of cells with a compound that has an impact on cell fitness or cell proliferation. And as a consequence of that treatment, most of the gene trap mutants will die, as well as the wild type cells, if there are any in that population. However, there will be a few knockouts that become resistant to the treatment with a compound. And then we can actually exploit the fact that we know the sequence of the gene trap cassette, and we can PCR out of that gene trap cassette and identify adjacent regions by next generation sequencing. So if you like, in one experiment, we get a comprehensive view of the mutants that confer resistance to compound treatment. So let's look at a first example of what such a screen can deliver. Here we picked an example that is a BCL2 inhibitor. It's a compound called ABT737. I'm not going to go into the details. Suffice it to say that that compound triggers apoptosis because it inhibits a survival signal, that is the protein BCL2. And you can then ask, what can we learn about the mechanism of action of this drug by running the experimental workflow that I just described? So we're treating cells with ABT737, and we're asking which mutants become resistant. Now let's have a brief look what we can expect from such an experiment. First of all, we cannot expect, so this is the pathway, if you like, from the BCL2 inhibitor to cell death. And we cannot expect to find BCL2. Why can we not expect to find BCL2? Well, the prerequisite for that experiment to work is that BCL2 inhibition needs to affect cell viability or cell fitness. And so as a consequence, usually the drug target, in that case BCL2, cannot be knocked out. The knockout of the drug target is in fact usually lethal because it phenocopies the compound treatment. So we would not expect to see BCL2. Now, if there are two possible ways, two redundant ways by which apoptosis can be triggered, we do not expect to see the components or the proteins Y1 and Y2. Why do we not expect those components? If there are two redundant paths by which the cell can succumb to cell death in response to that compound, then inactivating one of them will not be sufficient to prevent cell death. So Y1 and Y2 will not be seen. What we can find, though, is protein Z in that schematic. So we can find proteins involved in the response to the compound that are non-redundant effector proteins. So let's look at the results that we generated for the BCL2 inhibitor. This is a typical output of such an experiment. Let me guide you through that. On the x-axis, we're listing the genes that were identified in that screen. So we typically, as we're dealing with a very large population of cells, we're identifying several thousand independent insertions. However, we're then scoring each of them, and we're asking, are there any genes that accumulate more than one or two or three such insertions? And as you can very clearly see, there are two genes that stick out in that screen. One is a gene called BAX, the other one is a gene called NOXA. BAX was identified with 51 independent gene trap insertions, and NOXA was identified with 39 independent gene trap insertions. That means that there were essentially 51 clones in the initial population, all of which became resistant to treatment. And because that is a very unlikely event to occur by chance, the p-values associated with this particular experiment are just stellar. They're very, very low. It becomes very, very unlikely that such an enrichment is observed by chance. And this is really the key selling point, I would say, on that technology, is that the results that it delivers are really of a different quality than what you would expect, let's say, from RNA interference screening, where it was usually very difficult to distinguish between true signal and noise. Here, it's a very clear picture. And so if you then go back to the plot that I showed initially, this is just to remind you that we cannot really find the target, but if you had not known that VCL2 was the target in that particular screen, and you had identified BAX and NOX-A, by that resistance signature, it's usually possible to formulate a reasonable hypothesis as to what the target of that compound is, and it's usually useful information that helps you understand the mechanism of action of your drug or drug candidate in question. Let's look at a second example of how that technology performs. Oh, yeah, and then of course, if you want to go back to the problem that I presented initially, knowing about the fact that loss of function mutations in BAX and NOX-A give rise to resistance to that particular BCL2 inhibitor would, of course, enable you in a clinical trial to then look at patients and see whether there are any natural variants or any somatic mutations present in these patient populations. And if they are, you would potentially stratify patient populations and exclude those from your clinical trial, with the obvious consequence that you would increase the success rate of your clinical trial. Let's look at a second example of how that technology. The second example that I would like to share with you is on a Survivin inhibitor, YM155. This is a very interesting molecule that's currently in several clinical trials, including one against non-small cell lung cancer and one against metastatic breast cancer. And the screen was done in a very similar fashion. You take the haploid cells, you mutagenize them with a gene trap, you treat with a compound, and you ask, what are the genes that confer resistance to the compound? Here again, this is a slightly different representation of the results, but it's very obvious, even though you may have never seen that circus plot, this is what this plot is called, that there is a single gene conferring resistance to compound treatment, and that is a gene called SLC35F2. And again, the remarkable thing about these screens is that gene was identified with 122 independent insertions, and as a consequence, the p-value is really extremely low, which gives you the greatest possible confidence that this result is real. And so with this particular gene, this is actually taken from a publication. The authors went a step further, and they looked at the approach that I described to you earlier. They turned their attention to the Cancer Cell Line Encyclopedia. This is a famous panel of cell lines for which we have a great knowledge and a lot of information at hand. And so they look at that panel of cell lines, and they organize them, if you like, by the expression level of SLC35F2. There are some cell lines that naturally have high levels. There are some cell lines that naturally have low levels. And then you can correlate the expression level of SLC35F2 with the sensitivity towards YM155. And as you see on the plot in the lower left, there is a great degree of correlation, strongly suggesting that that the results that you just obtained from this rather, let's say, artificial haploid human cell line translates very nicely into the Cancer Cell Line Encyclopedia that people frequently use to assess sensitivity and resistance towards drugs. And you can pick out two examples, from either end of the expression range. You can pick cell line called SW480 that has high levels of SLC35F2. As a consequence, this cell line is highly susceptible to treatment with YM155. The opposite is true for U7 cells that have a low level of that particular gene product, and they're highly resistant. So all in all, it would suggest that what you learn from that haplogenetic screen translates very well into other cell lines, and of course, ultimately into the patient. I'm just mentioning here that there are a number of screens published in the literature, and if you're interested, we're happy to assemble a little literature package for you. Screens were done for numerous compounds. Again, the common theme here is that all of these compounds need to have an impact on cell viability or cell fitness. That is the prerequisite for these kinds of resistance screens. Now, what can we do if the compound that we're studying or the compound that we have an interest in has no impact on cell viability or cell fitness? Well, in that case, the compound might have an impact on a transcriptional readout. And if that is the case, we can actually also imply reporter-based screening. And I want to just quickly illustrate how those screens are done. So here the assumption is that treating the cells with a compound will elicit a transcriptional response, and that we can measure that transcriptional response using RNA sequencing. And what we can then do, and that can be a quite distant, let's say, downstream effect. It doesn't necessarily have to be a very direct consequence of the compound treatment. But if that is the case, if your compound of interest triggers a change in, let's say, the global transcriptional profile, an example here could be, for instance, an HDAC inhibitor, then you could actually go ahead and look at that RNA sequencing data set, pick a gene that's strongly upregulated in response to treatment, and use CRISPR-Cas technology to engineer the haploid cells to express a reporter in the endogenous context, in the endogenous gene, to express a reporter that will light up once the compound hits the cells. Can we actually do this? Can we actually engineer cell lines bearing a GFP reporter? Just to demonstrate that capability, I plotted in two examples where we decided to tag two quite famous genes with turbo GFP. This is a GFP variant. And so the first gene we decided to tag is lamin A, and you can see very nicely that we get a nice staining of the nuclear envelope. And the second gene is a gene called TRS1, and that is a protein that resides on telomeres, and that's why we get this very nice dotted staining. Anyway, this is just to illustrate that we can tag endogenous genes in these haploid cells with GFP. And so if we're able to make that cell line, then let's have a look at how that screen would then be done. Again, we would start off with the haploid cells, and these haploid cells would already contain the GFP reporter integrated. We do the retroviral mutagenesis, introduce the gene traps, so that we end up with that mutant population of reporter cells. But these, in the absence of compound, as shown on the upper right, in the absence of compound, these cells would be GFP negative, because the gene that we picked is a gene that would only be expressed in response to compound. When we then treat the cells with our compound of interest, most of the cells would actually light up. Most of the cells would mount a GFP response. However, there will be a small set of mutants that no longer mount that response, and that set of mutants contains the mutants that we're most interested in. And in fact, that population will also include the target of the compound. What we can then do is very straightforward from a technical point of view. We can FACS sort the cells and isolate the GFP negative population, and then again use the gene trap as a handle to PCR amplify the regions adjacent to the gene trap and analyze them by next generation sequencing. Okay, so let's summarize these two parts. I've shown you how we can use haploid human cell lines for gene trap screening. There are two experimental workflows that I discussed. The first one is resistance screening. Here the major prerequisite is that the compound needs to affect viability and fitness of the haploid cells. It's relatively straightforward and it doesn't really require any engineering. I would argue that the results that we get here are really of unprecedented clarity and precision. And if you've done similar experiments with RNA interference technology, you'd be surprised about how clear they come out. And the major application that I see for that type of screening is to profile drugs mainly in the oncology space, but not exclusively, to identify their mechanism of action and identify genes that, if knocked out, confer resistance to the drug. The second area is photo-based screening. Here the prerequisite is that the compound needs to affect the transcriptional profile, needs to have some impact on transcription in the haploid cells. It does require a little bit of genome engineering because we need to make these GFP reporter cell lines. The clarity and precision, I would argue, is comparable to the previous workflow. And the major identification here is probably target identification, where we're dealing with compounds that that were derived or that were picked up in phenotypic screens, and where the drug target and the mechanism of action is not fully resolved. So let me summarize the major points on this. I think one of the important points here is that these screens are unbiased. The retrovirus gets essentially delivered to greater 98% of the expressed genes. That means that almost any gene that is expressed in these cells is targeted and can be identified from these screens. They're comprehensive, so we're looking at all the genes in parallel. The screens are very robust. We've seen that if these screens are run in different labs in parallel independently, they turn out the same results, and the results are of unprecedented clarity. And there are a number of high-impact publications that support that this is a very attractive technology. In the last 5 minutes or so, I would like to illustrate the way forward. You know, what would you actually do when you've identified a gene of interest? How would you actually move on from there? In that case, we would actually turn to CRISPR-Cas technology. Many of you might have heard about that technology. It's based on a bacterial endonuclease that's called Cas9, and Cas9 can be programmed by short RNAs to introduce a double-strand break pretty much at any position in the human genome and in the genome of other organisms as well. And so the beauty of that is that we can introduce these double-strand breaks, and we can introduce mutations pretty much at where we would like to introduce them. So it gives us a surgical tool to inactivate single genes in human cells. Let's look at one example of how this technology performs. Here we're targeting exon 1 of a gene called GSK3 beta. So we're generating a single GSK3 beta knockout cell line. We're isolating several clones, and we're sequencing these clones. And one of the clones that came out in that particular example contained an 8 base pair deletion, as depicted down here. And that 8 base pair deletion disrupts the open reading frame of the gene, and as a consequence, the protein sequence eventually runs into a stop codon. And if you take a couple of these cell lines that we generated here at Horizon in Vienna, you will appreciate that if you Western blot them, they're all complete knockouts. And it makes sense that they're complete knockouts because in every one of these cases, we're disrupting a coding exon with a frameshift mutation, and that's why these cell lines are really complete nulls. And in the meantime, we've actually assembled what we believe is the largest collection that is based on CRISPR-Cas technology. It covers knockouts for more than 1,500 human genes. These are really available off the shelf, meaning that we have clones that are characterized, that are sequenced in our freezers, and they could be delivered to you essentially within 5 or 7 days. Every one of these clones goes back to an individual guide RNA. It's sequence verified by Sanger sequencing, and we can usually provide more than 1 clone per guide RNA. We've also organized our collection in a certain way, and we decided to target various gene families and pathways on that slide that we already covered. Importantly, if your gene of interest is not yet covered, so if you would like to study the mechanism of action of a gene that's not in our current collection, we can make on-demand knockouts for any human gene in 10 to 12 weeks for a very economic price. And we usually interact with the customer either via email or through our web interface. We then design guide RNAs in-house, we produce the clones, we quality control them, and send them over to you by FedEx. And this is all possible because these haploid human cells are so amenable to genetic engineering that we could really design and define a process that allows us to produce these at unprecedented speed and throughput. So let me summarize the path of target validation, which as a follow-up. So once you have done an unbiased genetic screen, you would usually like to analyze candidate genes that came out of that screen. And so the next step in that case would be to generate single gene knockouts by the technology that I just described to you, and that is CRISPR-Cas technology. You receive at least 1 clone. If you like, you can get a second clone. The cell lines bear a frame shift mutation in a coding exon, and we believe they're highly useful. Why should you consider these models? We believe that they're a very quick way to test a hypothesis, and we're also providing the guide RNAs to you that we used, so that if you see something that is encouraging in our cell models, you can actually move on from there and design and generate more sophisticated models using our reagents. These cell lines are very affordable. They're verified through Sanger sequencing. We have what we believe is the largest collection available to date, and they're usually available very quickly, which many people find important these days.